Okay, here we go with Prokaryotes 3 for BIOL 110. We have a few different learning objectives. The top is our summary. We're going to talk about beneficial bacteria, symbiotic relationships. We're going to kind of cover symbiosis in a little more detail. We'll get outside of bacteria a little bit just because it's really cool and important as we talk about commensal, mutualistic, and parasitic relationships. And then we'll dive back into how they relate to bacteria. We'll talk about anaerobes, obligate anaerobes, facultative anaerobes, aerobes. We'll talk about nitrogen fixing, one of the most important roles of these things. And then we'll kind of end up with oxygen and how these things got so much oxygen on our planet. So, kind of like cockroaches, bacteria tend to get a pretty bad rap. Everyone's like, oh, bacteria, they're so bad, they're just gross. And are most bacteria harmful? The answer is an overwhelming no. We need bacteria so much. They do so many things for us. We have so many symbioses with them and very few parasitic pathogenic relationships with them. For example, nitrogen fixing, which we're going to talk about in great detail a little bit later, are one of the most important roles that we have with these, with bacteria. They basically make all life possible. Really important. Maybe another question: are they harmful? I hope not, because you have hundreds of species in your gut right now, and a bunch more all around you. In fact, probably almost everybody right now listening to this in our class, etc., has some pathogenic, meaning disease-causing bacteria in their gut right now. But they're probably being controlled in their numbers by other bacteria that are there that are slowing it down. There's also bacteria in your gut that provide you with vitamin K that we have no way of making ourselves, and we don't really get from much of our food, but this thing cranks out vitamin K, and we need that for blood clotting and other parts of life. So there's some extremely important bacteria. They're protecting us from all these negative bacteria. They're they're helping us out with vitamin K. There's another one we'll talk about a little bit later. There's so many good things that they do to break down things for us. And many of these live in these symbiotic relationships with us, along with other things that are really important. So when we talk symbiosis, we can talk about lots of different symbioses. You're probably familiar with some, like the clownfish and the sea anemone are a really good example. On the over here on the right, we have some sea fans and some brittle stars that help clean off and keep these sea fans clean and healthy, which they're filter feeders that help filter out stuff in the water. Here's a moray eel with little fish that are picking out its teeth. Moray eel could easily just chomp down those things, but they do so much good that moray doesn't want to do that. And we have three types of symbiosis: mutualism, parasitism, and commensalism. They're really important. Mutualism is both things get some help. Most symbiotic relationships we talk about, refer to, are mutualistic relationships. Parasitism, parasitic relationships, one derives a benefit, the other receives a cost. Like in the bottom here, we have a tick and a cat, right? The tick is feeding on the cat. The cat's getting nothing good out of the tick. The tick is getting everything good. So it's good for the tick, bad for the cat, right? Happy face for the tick versus happy and happy. Oops. Let's make that actually a smile. For the mutualism. And then commensalism is one is unaffected and one is benefited. Like barnacles living on a whale, they get a great benefit by a place to live and exposure to lots of water. They're also filter feeders, so they got their little filters out. The whale doesn't seem to be affected terribly by the barnacles. We don't know for sure. It could be a more complicated relationship than we are familiar. Here's another little video by Hank that we'll watch about commensalism.
Today, People all around the world are celebrating love and relationships, which means that this episode is all about bacteria. Specifically, the beneficial relationships called mutualistic symbioses between bacteria and animals. In nature, you can see lots of symbioses with the naked eye, like clownfish and sea anemones or bees and flowering plants, but there are also plenty between animals and bacteria that you would need a microscope to observe. And like, no offense to Beyonce and Jay-Z or whoever, but they might be some of the coolest relationships out there, because in exchange for a nice place to live, many bacteria give their animal companions superpowers, like immunity to poison or invisibility. So in honor of Valentine's Day, here are 5 of the coolest animal-bacteria partnerships. Unless you grow plants, you probably haven't thought a lot about aphids lately. They're the little insects that suck the fluids out of vegetation and transmit plant viruses, and although they're major pests, they're actually pretty impressive. There are more than 4,000 known species throughout the world, and they reproduce ridiculously fast. An aphid can have about 80 babies in just 10 days. In fact, while they can mate, most are essentially born pregnant through a kind of asexual reproduction called parthenogenesis. This makes aphids great at multiplying quickly and damaging lots of vegetation. But their diet also isn't that great for them. The juices they eat lack many of the essential amino acids they need to survive. You'd think that the response to this would be to eat a more varied diet, but the aphids found a more creative and lazier solution. Many have teamed up with bacteria that make amino acids for them. They're from the genus Buchnera, and they're passed down from generation to generation while the insects are still embryos. These bacteria produce some of the missing amino acids themselves, like tryptophan, and in other cases, it appears that molecules from both the aphid and the bacteria work together to synthesize the molecules. Scientists estimate that this partnership began up to 250 million years ago, possibly after bacteria living in aphids' guts made themselves indispensable, and now neither organism can live without the other. The bacteria allow aphids to survive on meals that would be impossible for other insects. And the aphids give the microbes food and a safe place to live. They have special cells called bacteriocytes that they use to house their microscopic buddies. This relationship is so successful that the bacteria have even jettisoned a large chunk of their genome, including genes for responding to environmental changes and for building strong cell walls. Because, like, if you already have a nice house and a stable environment, those genes are useless. Now, these bacteria are down to around 500 genes, while bacteria that live independently usually have at least 1,500. So the next time you hear about aphids going to town on crops or your friend's houseplants or my cherry tree, know that it is not the insects. It's a whole power couple. Competition for food can be fierce in the animal kingdom, especially in harsh environments like deserts. There generally isn't a lot of food to go around, and the stuff that does exist, like plants, doesn't really want to be eaten. Desert plants often have defenses like spikes or poison that keep most would-be diners away. One example is the creosote bush. It contains a toxic resin that can cause severe liver and kidney damage. Except some animals seem to be more or less immune. For decades, scientists have observed desert wood rats voluntarily eating the plant. By itself, this isn't that weird. Some animals can resist toxins. That's like normal evolution stuff. What was weird was the discovery published in 2000 that not all desert wood rats, even within the same species, had this immunity. Wood rats living in Southern California's Mojave Desert, where the creosote bush grows naturally, were able to eat a lot of the plant without getting sick. But wood rats living farther north in the Great Basin Desert, where the bush doesn't naturally grow, got sick and lost weight after being fed the plant in experiments. It took more than a decade before researchers figured out why. In a paper published in 2014 in the journal Ecology Letters, scientists revealed that it wasn't the rats themselves that were different. It was their gut bacteria. When Mojave wood rats were treated with antibiotics, their superpowers disappeared, and they could no longer eat the creosote bush without getting sick. On the flip side, when the Mojave gut microbes were transferred to the Great Basin rats, the northern wood rats gained the ability to eat the plant. Scientists still don't know exactly what type or types of microbes are necessary for this immunity. Given that creosote bush resin contains several toxic compounds, it's likely that many microbes work together to protect desert wood rats, rats, and that they're passed around through coprophagy, a fancy word for eating poop. But whatever they are, they're doing a great job, and in exchange for helping the wood rat stay alive, the gut bacteria are rewarded with a steady stream of nutrients whenever that rat eats food. Life somehow manages to exist in all kinds of bizarre places, including the bottom of the ocean. When scientists first observed deep-sea hydrothermal vents in the 1970s, they were amazed at the density and variety of life clustered around them. In particular, the deep-sea clams they found, now called vesicomyid clams, were much larger and more numerous than they had expected. After all, most clams are filter feeders, getting their nutrition by straining tiny organisms like plankton out of the water, and down at the bottom of the ocean, clam food is in short supply. Today, we know that vesicomyid clams manage to live down there as deep as 6,800 meters with the help of symbiotic bacteria. The clams have extra large gills containing lots of bacteriocytes, which house microbes that oxidize the sulfur pouring out of the hydrothermal vents. The bacteria harness the sulfur's energy to support both themselves and the clams, and in return, they get a safe place to live. Kind of like with the aphids bacteria, these microbes ditched the genes related to cell structure and movement, and they haven't been found anywhere outside the clams. Also like aphids, these clams acquire their bacteria before they're born. Bacteria from a mother clam is housed inside her eggs, so that when the offspring hatch, they're already equipped with the partners they need to survive. Most clams, and most other living things, could never survive at the bottom of the ocean so far from sunlight. But with the help of these special bacteria, this clam has carved out a niche in an incredibly competitive ocean. Another superpowered ocean dweller is the blue-ringed octopus. This animal is super cute. It weighs less than 30 grams and would probably fit in your hand, but I wouldn't recommend picking one up because it is also one of the most venomous creatures on the planet. Its venom contains a potent neurotoxin called tetrodotoxin, or TTX. It's about 1,000 times more toxic than cyanide, and it works by blocking the sodium channels on nerve cells. This stops the cells from firing, which means they can't tell muscles to move. That means like the muscles that move your lungs, and that would lead to respiratory paralysis and death within minutes. Which seems kind of like overkill when you consider that the blue-ringed octopus mostly uses this venom on tiny crabs and mollusks. But that's beside the point. What's especially interesting is that the blue-ringed octopus doesn't produce its own venom. Instead, some researchers think it's produced by symbiotic bacteria in the octopus's salivary glands. That being said, there is some controversy about this, and not every study has been able to grow TTX-producing bacteria from those glands. But many scientists are on board with the idea. Scientists are good if you get poisoned yourself, so like other animals that use TTX, including pufferfish, the blue-ringed octopus also has evolved resistance to the toxin. Among other things, its nerves have a different type of sodium channel that TTX doesn't disrupt. So these little animals are able to go about their lives paralyzing others while staying themselves quite safe. Finally...
Okay, sorry about that. It had a little malfunction in the middle of Hank's discussion. So I'm just decided to cut it off. You can just watch it. You're on the link is right there. It is available on the notes. I do encourage you to watch it. There will be a bonus question dealing with that video. I'm not going to ask a lot of details, but you should pay attention. It's really cool stuff. A couple things that he said that I just want to clarify. Buchnera is called Buchnera, not Buchnera. It's the bacteria that lives inside aphids. It also is passed vertically from mother to offspring. The bacteria migrate to the eggs prior to her laying her young, and that's how they get passed on from one generation to the next. Okay, so we have a bunch of different types of symbioses. Mutualism, when both species benefit from the relationship. Here's a couple of cool ones that we can talk about here. You can see them right here. There's some ants. This is the longhorn acacia. These needles are actually empty, and the ants live inside of that. And they make a little nest in there, and they actually protect the whole plant from other things that might attack it. If any other insect lands on there, those ants are going to attack it and really mess with it. So that's kind of a nice little mutualism between the plant and the ants. In this case, the plant does not like the ants because the ants actually protect the aphids, which are feeding on the plant. So the aphids are feeding on the plant. The ants are getting the mildew from the aphids, and they really like that. And these videos are about mutualisms. You can watch them if you like. I strongly encourage you to. They're really cool, but I'm not going to play them directly through this PowerPoint. You can kind of check them out on your own just to see some cool videos about some mutualisms. All right. Another one is commensalism and also parasitism. We've already talked about commensalism. Sometimes commensals become pathogens or become parasites. That's what we call an opportunistic infection. We'll talk about this a little bit later with another one, but sometimes you have a bacterium that's on the outside of your body. It's really good. Maybe it's helping you out, being beneficial, but if you get a cut, it could infest into your blood and cause a pretty serious infection that way. And that is not an uncommon thing to happen. Tetanus is like that. Tetanus is not something that likes to thrive as a pathogen, but when you get a good cut, then it will infect in there and cause some serious problems. That's why you get a tetanus shot every 10 years. Or if you get a really bad cut and something, it gets really dirty, they say, if you got a tetanus shot, like, well, let's give you one, because you definitely don't want to get infected with that kind of a bacterium. And then parasitism is when one derives a benefit at the cost of the other. All right, so parasitic prokaryotes, which they're designed to be pathogens, like these disease-causing, like Clostridium, they're parasitic all the way, or all the time, excuse me. Let's go to the next slide here, so we can talk about these exotoxins and endotoxins. So parasitic prokaryotes that carry the disease are called pathogens, like Clostridium botulinum, which we talked about in the last lecture. That secretes an exotoxin, which are proteins that are secreted by a prokaryote. So remember, as it goes into sporulation, it excretes this protein that turns out to be a neurotoxin, and it's pretty bad news. It's probably designed to keep that thing safe when it goes into sporulation, doesn't get eaten by stuff. The endotoxin are surface protein components of the outer cell membrane and some gram-negative bacteria. So they're kind of built into the bacteria. If you get an infection with a bacteria, it has an endotoxin. Exotoxins, we already talked about botulism. Cholera is also an exotoxin pathogen. Salmonella typhi, that causes typhus, that's an endotoxin that counteracts the human host defenses. Not very nice. All right. And we already talked about our skin bacteria being commensals, but then turning into pathogens or parasites as opportunistic infections. Other parasitism, other notes about parasitism, one actually kills the host all the way. So I have here just some cycles from the CDC of different parasitic interactions. This one up here is just a parasitoid wasp. They mate, and then the wasp finds a caterpillar. It lays its eggs on the caterpillar, and they kill the caterpillar and develop inside of it, and eventually kill it, and then hatch out as an adult from eating out the inside of the caterpillar. That's a parasitism. Here's more parasitisms that you can get, like a tapeworm. A tapeworm will live inside your intestine. It'll shed out little, they're called proglottids. They'll get eaten by a rodent or by a pig or some kind. And then when you eat undercooked meat, you get that back, or you get the original. I mean, it's the first time it's in you. And we have other ones here that are really cool. This video is really fun. It's actually kind of funny. Wow, it's a terrible arrow. It's by this guy named Z. Frank. It's about ants, all these different ant mutualisms and ant things. And then it talks a little bit about these fungi that infest ants and then zombify their body and command them what to do, and then they grow out of the head of the ant to shoot off spores. You can watch that one on your own too. It's well worth it. It's really funny. There's a little bit of language in it, so I'm not going to play it here. So Be cautioned if you would not like to hear that. Don't click on it, but there is some funny things going on in there, and there's some scientific accuracy. Okay, and then we've already talked about commensals quite a bit. I'm not going to hit this anymore, but barnacles or the remora fish on sharks, these are good examples of commensals. So, the relationship can change. Sometimes a commensal or mutualistic can become parasitic. We've already talked about how this could happen on your skin. There was actually a study at Penn State looking at these synthetic turf fields, and could they carry more prevalence of staph infections than just a regular grass field. A regular grass field is probably better because it's a biological environment. There's lots going on there. There's plants and soil, and then lots of bacteria, and they're kind of all living in harmony together, and that's really good. When you get out to this field like this, it's not so good. You're getting rid of most of the biology, and so then bacteria only can kind of live on there, and they have a slightly increased rate of MRSA infections. At least this is what was being studied. I think they found it did not have a dramatically high increase of staph infections. There was a few, but MRSA stands for methicillin-resistant Staphylococcus aureus, and that causes these staph infections, which are hard to treat because they're resistant to antibiotics already, which is not good. Okay, so some cool things that have happened is disease repression or disease suppressive soil. Okay, so underneath the plant, there's the soil around that's called the rhizosphere, and it's a really complex web of nutrients, microorganisms, macroorganisms. You got insects, you've got bacteria, you've probably got some fungus in there, you've got your plant roots in there, lots of bacteria. I already said bacteria. You've got all sorts of things that are very complicated. Sometimes if a soil is attacked by a pathogen, then bacteria that fight that and fungi that fight that will grow stronger. They will help prevent future attacks by those plants. And so some researchers decided that they should test this in the Netherlands. It was a pretty cool research idea. They found a field of beets where the beets have been attacked by a fungus. So it's a pathogenic fungus. You can see here on the right side of the right image, beet that got attacked by this fungus. Pretty bad news. So the field had this attack. So they took soil from the field that had been attacked and then took soil from the margin of the field. The margins of the field were outside of the rhizosphere. There should be pretty much the exact same soil, right? You don't have all these confounding variables in there like, oh, is it the same acidity of the soil? Is it got the same water? Does it have the same, all these different non-biological things that could be taking place. So you take the soil that's right next to the field, but should not have had the rhizosphere of the plant that got attacked by the fungus, and see if stuff in that rhizosphere will actually fight back against invading fungus again. And so the researchers gathered field and margin soil. They had 5 treatments, 4 pots for each treatment, and 8 plants for every pot. So they were doing quite a few, looks like 32 plants for each one of these 5 treatments that they had here. And you can see here the results, okay? So what they have here, they have the soil treatment, right? And then they have the percentage of seedlings with the fungal disease. You need to think about which of these is the dependent variable and which is the independent variable. If you don't remember those from typical experiments, look them up. Which one causes things, which one affects things. Another way to kind of ask that. So we have disease suppressive soil. That's just soil from the field. Soil from the margin of the field. So from the around the outside, right? We got our field here. The disease suppression soil. We got the margins out here. Grab some soil from there. We'll color that This is margin soil, right? Soil from the margin of the field plus 10% disease suppression soil. So just a small amount of the disease suppression soil. It's kind of hard to say. Disease suppression soil heated. They should just call this field soil because they don't know. The whole experiment is it disease suppressive or not? They should call it field soil. And field soil that was heated to 80 degrees C for 1 hour. This is the percentage of seedlings with fungal disease. All right? So in the field soil, they had 3%. Right? That's very, very little. Looks like they probably had one because they did 32 plants. So they had one that died. Soil from the margin, 62% of the seedlings had the fungal disease. So 2/3, a lot of them. Adding just a little bit of soil, you have to drop it down to 40%. Adding a little bit of the disease the field soil. Gosh, I hate that. Take the field soil and heat it up for an hour. And you had 30% with a fungal disease. Heat it to 80 degrees C for 1 hour. You had 80%. So if you take that good soil, obviously this field soil is the best soil, right? If you take that and you heat it up, what does that do? Why are they heating it up? What is that showing? Well, that is showing. Think about it for a second. Maybe push pause and say, what is that doing? If you have live things in there and you heat it up, you're killing them. 50 degrees C for an hour is hotter than just about anywhere on the planet. So that'll roast that dirt. 80 degrees C for an hour for sure is. I don't even know if the hottest places, probably in the Sahara, maybe in Death Valley, you can get 50 degrees C. That's really hot. It's ridiculously hot. So looks like the field soil is disease suppressive, and it's something live that's doing it. And if you kill it, then it's no longer that suppressive. In fact, if you kill it, it's worse than the margin down there. The disease suppressive soil heated 80 degrees is worse than the soil from the margin of the field. So, and here's 3 questions you need to ask yourself and see if you can answer. I'm not going to give you the answers. Not that hard, but what hypotheses were the researchers testing in this study? What is the independent variable, the cause, and what is a dependent variable, kind of the effect? Okay? Out of we have 2 columns here in our little chart here. One's the independent and one's the dependent. Okay, going back to humans in our community, really a whole ecosystem. We are born sterile, right? We don't have any bacteria inside of a fetus, but then we start to acquire bacteria through food and contact. And everybody, turns out, has hundreds, most people over 1,000 species of bacteria when you look at the whole bacterial community around us. In 2012, they had the Human Microbiome Project, tested 242 healthy individuals. Each had about 1,000 species. And most people were actually carrying pathogens without being sick because the other bacteria were helping fight. Here's an interesting thing. They noticed that C-section versus vaginal birth had different bacteria. What does that say? What does that mean? Obviously, some bacteria picked up during the birthing process. That wouldn't be happening through a C-section. Complicated stuff. Other complicated relationships. Some termites have protozoan microsymbionts with microsymbionts inside them. So what we have here is we have these termites right here that they need to eat, and they eat wood. Wood is terrible. It's impossible to break down. So they have these protozoans that help break that down. But the protozoans actually need to have these bacteria inside of them that help break down what the termite is eating, right? They can't do it just by themselves. You don't need to memorize this. It's very confusing, very, very confusing chart. People are experts in termites and bacteria and symbiosis would know this kind of stuff. But you don't need to worry about you knowing it. Just knowing how complicated it is. Bacteria living inside protozoans, living inside termites. Going to mealybugs. Mealybugs are closely related to aphids. They also have endosymbionts. They actually have bacteria that live within bacteria that are living in the mealybugs. In Hank's video, hopefully you were watching, he was talking about the bacteriocytes. Those are cells that hold bacteria. So let's see if I can kind of draw this out. So we'll say here is a bacteriocyte. Okay? Inside that bacteriocyte, in the mealybugs, it's called Tremblea, are a bunch of bacteria. Named after Tremblay. Buchner was named after Buchner. That's why we know the right way to say his name. He's German. Inside of those bacteria are other bacteria. These are called Morinella. All right, so what we're looking at here, this is one bacteriocyte with blue bacteria inside of it and red bacteria inside of those bacteria. That's bugs within mealybugs was the catchy little name that they gave the paper. And they named these things Morinella after Nancy Moran. When Hank was talking about the scientists that discovered the amount of time that Buchner had been living inside of the aphids, he was actually talking about Nancy Moran. She's kind of the world expert on Buchnera. And so when they found these bacteria inside of mealybugs, they were named after Nancy Moran, which is pretty cool. Okay, so nitrogen fixation. This is a really important thing. This figure is not so good. It's kind of difficult. It's kind of difficult to follow, to be honest with you. But nitrogen fixation is one of the most important processes in the plant. There's nitrogen all around us. 80% of the air or more is nitrogen. But we can't access it. There's nothing we can do with it. But nitrogen-fixing bacteria can. So down here on this plant, on this legume, right, it's got nitrogen-fixing bacteria. And they can be nitrogen-fixing bacteria in the soil, then get it changed to ammonia, and then ammonia can make it into plants. Animals eat the plants. We eat the animals or the plants, hopefully more plants than animals. And that's how we get all our nitrogen, through the food that we eat. And we need that nitrogen. We need it for so many different things, right? So we need to get it from the plants, which get it from the bacteria, which are attached to other plants. It's a very complicated thing. And then fungi will get it from all the dead organisms. They'll break it down and they'll actually put it back into the soil. And sometimes they'll end up back in the atmosphere in this big nitrogen cycle. Very cool. This figure is kind of confusing, and I'm sorry about that. Some of those nitrogen fixers, the rhizobia and the legume system. So the rhizobia are the bacteria. They're living down in the soil. It's really cool. So here is a legume root, right? That's what we're looking at right there. At the proper time, they actually will release. Let's get a different color for that. They will release basically like a scent, a biochemical, and it will tell all these little bacteria, make them green this time, oh, this plant needs nitrogen. And if we go there, they'll make these little nodules for us. On the plant, here's the nodules right there, right here, right here, right here. And when we live in those nodules, then all we got to do is take nitrogen out of the soil. And if we take nitrogen out of the soil, then the plants will give us back some sugars and things so we can survive. So it's a nice mutualistic relationship going on there. And that all of life kind of depends on this one, or on all the ones like it. The rhizobia that will come up, they'll latch onto the roots, they'll provide nitrogen in exchange for some sugary goodness from the plant, and everybody's happy, including us who are going to eat the plant and then get that nitrogen, and so on and so on and so on. Another one that's great, that is so useful for people doing agricultural, especially in South America and Asia. I know this one is in Asia. I don't know if it's in South America, though they do eat a lot of rice down there. So in this case, we have a cyanobacteria. Remember, that's the cyanobacteria is the one that does all the photosynthesis. Actually, we'll talk about that in a minute. Sorry, getting ahead of myself a little bit. The cyanobacteria and the algae can fix nitrogen. Okay, so one particular, the Anabina colonies they're on an aquatic fern. So it's a cyanobacteria and an algae that lives on an aquatic fern, and it puts nitrogen into the water in rice paddies. It's a natural fertilizer that just fertilizes all these rice paddies. They typically don't require a huge amount of fertilization for that reason. Okay, so let's talk about some global impacts of these prokaryotes. We've talked about early Earth a lot, right? Really hazardous, terrible atmosphere for us anyway. So about 3.5 billion years ago, the whole planet was anaerobic. There was no oxygen. Oxygen was basically toxic for everything that was living. Early on, these prokaryotes were chemoautotrophs. They make their own energy from the atmosphere without oxygen. They probably got energy from hydrogen sulfide, like some archaeans still do right now. Frank and I disagree on which ones were first, the bacteria, the archaeans, so we're not going to worry about that. We don't know for sure. But then, somewhere about 3 billion years ago, give or take, the first cyanobacteria that do photosynthesis came about, right? And they would use water as an electron source. So when they would do this whole process, like all photosynthesis, they release oxygen. The spike in oxygen changed the landscape, I like this, into a poisonous hellscape for life. It was terrible. Terrible, terrible, terrible. You can see here, this image is good. It kind of goes around backwards. I don't really love it. But it's going along. This is time going this way. And then we see a little spike in oxygen there, and then it shot up right here, and then got up to be 1/10. And actually, that doesn't show it on here, because this is 1.5 billion years ago. But probably about 400 million years ago, it actually shot up even higher, and then went back down to where it is. That's when there was huge insects and things like that. There was really, really high oxygen at the time. This one is a good video. We're going to watch it. It's not terribly long. But we're going to talk, we'll talk about how oxygen almost killed all of life. It's a cool one from PBS Eons. But anyway, we have obligate anaerobes. They need oxygen. Obligate aerobes need an oxygenated environment. We are obligate aerobes. Obligate anaerobes, oxygen is toxic. So like the Clostridium botulinum that we talked about, that's an anaerobe. Oxygen is toxic. Lots of bacteria still survive that are toxic if there's oxygen. And then some are facultative. Facultative means it can kind of go either way. They alternate their oxygen needs. Okay? So I'm going to pause this.
This episode is supported by Squarespace. Are you like me? Do you think oxygen is pretty great? Of course you do. But what if I told you there was a time when oxygen almost wiped out all life on Earth? This is the story about how too much of a good thing can ruin everything for everybody. Let's start 3 billion years ago in the Archean eon, when the world was a place you'd never recognize, or even be able to survive in. Back then, only even be able to survive in. Back then, only 2 or 3% of the planet's surface was dry land. The rest was covered in oceans, and they were full of iron. Because of all that iron, the oceans were probably not blue, but green, and not because of algae or other life, but because of rust. You might think of rust as being brown, but green rust is a thing too. It forms where there's a lack of oxygen, and back in the Archean, oxygen was in short supply everywhere. So in the oceans, iron reacted with hydroxides and elements like sulfur and chlorine, covering much of the world in green rust. Above the green oceans, the atmosphere was mostly nitrogen, as it is today, along with water vapor, carbon dioxide, and methane. But again, very little oxygen, which was fine at the time because microscopic life on Earth back then was probably anaerobic. Then, about 2.8 billion years ago, some new kids showed up, photosynthesizers, but not the kind that we're used to. These were bacteria, like the familiar blue-green cyanobacteria, or maybe even a kind of purple microbe known as halobacteria. Either way, they used the sun's energy to convert CO2 and water into food, releasing oxygen as waste, which was okay for a while, but not for long. Because when these little microbes started farting out oxygen, it changed the chemical balance of the entire biosphere and eventually altered the face of our planet. First, within about 200 million years, the oxygen from these new bacteria began reacting with the iron in the oceans. This turned the ocean from a mellow green to a deep blood red, as the seas began to fill with iron oxide, what we know as rust. In fact, you can still see exactly when and where this happened in strata known as banded iron formations, where huge swaths of rust settled out of the oceans to form layers of red-brown rock. But beyond making the water rusty, rising oxygen levels also shifted the balance of power among living things. Anaerobic microbes started to die off, as they were basically poisoned by the oxygen. Meanwhile, the newer, more efficient photosynthetic life began to spread. And things changed even more when the oxygen in the ocean left the water and entered the air. For the first time in Earth's history, large amounts of free oxygen started building up in the atmosphere. And this is what scientists call a game changer, because then the climate started to change too, drastically. As the photosynthetic bacteria kept spreading, they used up more and more of the carbon dioxide in the atmosphere, and CO2 is a greenhouse gas, so as its levels went down, so did the temperature. On top of that, oxygen also started reacting with the methane in the atmosphere, taking it out of circulation. With greenhouse gases at an all-time low, our planet started to see the first major ice ages in its history. Temperatures dropped so drastically that it triggered a global glaciation, and our once balmy water world became shrouded in ice. There may have been several pulses of these icy events, but by far the biggest of them came to be known as the Huronian glaciation, and it lasted for about 300 million years. Between these huge changes in climate and the radically different chemistry of the planet, most of life on Earth was pushed to the brink of extinction. And this included the new guys, the photosynthetic bacteria that were pumping out all the oxygen in the first place. Because remember, they needed CO2 to survive, so they were basically being suffocated by their own waste. Which, when I say it out loud, that's gotta be a terrible way to go. We don't know the full extent of the damage, but the effects of this whole episode, sometimes called the oxygen catastrophe, is considered one of the biggest extinction events in history. But you know how this movie ends, right? By the time the Huronian glacial period ended about 2.1 billion years ago, some life remained, and the life forms that survived inherited a better, more hospitable planet. Oxygen was now abundant in the air and water, and a new thing also appeared in the atmosphere, an ozone layer. This coating of molecular oxygen helped block dangerous ultraviolet radiation from the sun, and suddenly new things became possible. From there, well, you know what happens. Life became more complex and awesome and wonderful, and here we are. It seems strange that bacteria could end up covering the whole world in ice. And who'd have thought oxygen would be responsible for wiping out almost everybody? But it just goes to show you that sometimes things gotta get worse before they get better.
Okay, just a little bit more here. Sorry, I'm trying to do all this from home, and I just keep running into issues like my daughter's AirPods picking up and taking the sound away from her video. Okay, so a real life example. That's kind of cool. So we already talked about the number of species being over 1,000, but the human gut has up to 100 trillion bacteria. That just alone in your gut is 3 to 10 times more cells than a whole human body. Now, these bacteria, of course, are much smaller than our cells, but our cells typically have probably, depending on the size of the person, the age and everything, usually about 10 to 30 trillion cells. So it's a lot more cells just living in your gut. They provide metabolic capabilities we've not evolved, such as Bacteroides, teomicron, whatever, close enough. This is actually really helpful for breaking down big sugars. There's a lot of polysaccharides we end up eating that we can't break down. Probably would be good, actually, if we didn't have this bacteria in some ways. We didn't realize how good it was until we realized how much it did for breaking down these polysaccharides. One of the reasons we have so many different species, in fact, some have called it the most diverse piece of real estate on the planet. The most diversity happens just inside of our gut. We eat so many different kinds of foods, so many different things we eat constantly. So we get all these different bacteria that live in there. Anyway, this was really, really good. We thought it was just a commensal. Now we kind of think it's probably a mutualism. They are bad, though. They can turn into a pathogen. So, can be commensal maybe, could be a mutualism maybe, could be a pathogen. This is why if you have surgery on your abdomen, if these things get loose and they get into your stomach or get it out of your stomach into your blood and things, they're really, really bad, and they're antibiotic resistant for the most part. So you got to be really, really careful with these things. They're good. They're either like really good, doing nothing at all, or really, really bad, right? I kind of already answered all this. Okay, so that does it for Prokaryotes 3. I'm going to put this up right now. Enjoy the test tomorrow. That's my next thing I'm going to do today. And we'll see you guys soon.